First, a calcium loading solution is prepared, warmed, and bubbled. The urinary bladder is dissected out, opened, pinned out flat, and a strip of detrusor is taken. The detrusor strip is added to the loading solution, incubated, and rinsed. Pinned out flat, the calcium fluorophore within the tissue is excited by laser light and the emitted light captured. Resulting movies consisting of individual frames are analyzed by comparing successive frames. The background is subtracted, the cell edge threshold determines where events can be found, and a ratio between successive frames is taken. If the remaining events match predefined criteria, they are highlighted and labeled.
Hello, my name is John Young from the laboratories of Keith Brain and Tom Kinane at the Department of Pharmacology, the University of Oxford.
I'm Rob Amos, also from the Brain and Kinane labs.
And my name's Keith Brain. Today, John and Rob will show you a calcium imaging technique in smooth muscle. We use this in our laboratory to study the release of neurotransmitter from autonomic nerves and its effect on smooth muscle.
So let's get started.
The calcium indicator Organ Green 488 BAFTA-1 AM comes in 500 microliter vials containing 50 micrograms of powder. To prepare the calcium indicator, add 40 microliters of pluronic F127 solution to the powder and shake gently for 30 seconds. After that, add 460 microliters of physiological saline solution, PSS, and shake gently for another 30 seconds. The final solution, 10 micromolars of Organ Green 488 BAFTA-1 AM, provides 4 aliquot of 125 microliters each. Avoid exposure to light and store the solution at -20 degrees Celsius. Prior to dissecting the tissue of interest, remove the calcium indicator from the freezer and thaw at room temperature. While the calcium indicator is thawing, place 1 milliliter of PSS in a test tube in a water bath at 33 degrees Celsius. Secure a bubbler into the test tube with a bung, or you could also use parafilm, and then bubble with a 95% oxygen to 5% carbon dioxide mixture at a rate of around 1 bubble appearing per second. Dissect the smooth muscle organ from the animal. In this case, it's the urinary bladder from a mouse. Place the organ in about 50 milliliters of PSS to wash it. After the tissue has been washed, carefully remove the serosal adipose tissue and connective tissue. Dissect open according to the smooth muscle organ type. For the urinary bladder, open longitudinally from the bladder neck, the caudal end, to the top of the dome, the cranial end. Prepare 4 strips 6 to 8 millimeters long and 1 to 2 millimeters wide along the dorsal surface of the detrusor, cutting in the direction of the dominant muscle bundles. Place the dissected tissue in the pre-warmed and bubbled PSS. To load the calcium indicator, add 125 microliters of 10 micromolar Organ Green 488 BAFTA-1 AM to the tissue in the test tube and give it a little shake by hand to mix. Return the tube to the water bath, resume bubbling, and cover the water bath with a sheet of foil. The time it takes for sufficient tissue to take up the calcium indicator varies with the size of the tissue and the thickness of the smooth muscle layer. For example, for the mouse detrusor and rat anacoccygis, it's 70 minutes, whereas for the rat detrusor and mouse vas deferens, we incubate for 120 minutes. Once the tissue has been loaded with the calcium indicator, it can be prepared for microscopy. First, rinse the tissue in bubbled PSS for 10 minutes. Next, mount the tissue in a Silgard-lined preparation dish, stretching it slightly to form a flat sheet. Pin the tissue using short lengths, for example 2 mm, of 25 to 50 micrometer diameter silver wire and insert the pins at an angle. Avoid using pins that are any longer than a few millimeters in length, as they may scratch the objective. Careful pinning of the tissue is important for minimizing spontaneous movement of the tissue and to facilitate the location of a suitable site for imaging. After the tissue has been pinned, position the preparation dish on the microscope stage. Be careful that the PSS does not escape from the Silgard-lined area of the dish, as this may lead to a large area being covered in PSS when the superfusion begins. For demonstration purposes, the following steps will be done in the light. Please note that these experiments are normally performed in darkness. Switch on the pump in order to superfuse the preparation with PSS. A rate of 100 milliliters per hour is often used. Switch on the heater unit. Place the inflow and outflow tubes and temperature probe on the preparation dish, affixing each to the metal strip by the magnet on its base. The height of the tip of the outflow tube will determine the depth of the PSS. Be careful to ensure that the outflow is actually removing PSS, as it sometimes does not initially. Set the temperature on the heater unit to reach the desired temperature of between 33 and 35 degrees Celsius. Allow the tissue to equilibrate for at least 10 minutes. With a low power objective, 10 or 20 times, use epifluorescence or exciting with a blue light to view the smooth muscle and identify a suitable region to monitor. Since exposure to excitation illumination will photobleach the indicator, avoid using for more than a few seconds at a time. Avoid sites with bright structures because they may cause a high number of false positives in the image detection algorithm. Try to pick a site with minimal movement and a large number of smooth muscle cells in the field. Once you have selected the site to image, switch to a higher power objective, such as 40x. Rotate the stage or the optical axis so that the smooth muscle cell or cells lie horizontal, that is, parallel to the rapid scan axis. The smooth muscle cells are now ready for imaging by confocal microscopy. In this protocol, we use a Leica SP2 upright confocal microscope. Close the pinhole to one airy disc. Set the laser power between 25 and 35% on the acousto-optic tunable filter. This value is a trade-off between brightness and photo bleaching. To prepare for data analysis, download and install Image J. Download the platform-specific version and then update to the latest version by downloading the latest imagej.jar file to replace the old file. Next, download the Excel writer jar file and install into the plugins jar directory. Copy these files into the plugins directory. Install each of these scripts using the plugins, macros, install function of ImageJ. The software for the Leica SP2 confocal microscope that we use, called Leica LCS, saves individual frames of each series as a separate file. To reconstruct the series, make sure that all the frames to be reconstructed are in a single folder, for example, data folder TIFFs. Then, select the Leica SP2 stacker macro from the Plugins Macros menu. Select the root directory, in this case data folder. Select the desired folder from the drop-down list, in this case TIFFs. Select an output directory or generate a new one, for example data folder stacks. The script will then reconstruct series from individual frames. To correct for movement, we use a freely available algorithm that aligns or matches frames within a series. Download StackReg and TurboReg. Copy the files for each into the Stacks folder. Load a stack to be processed, and then move to a frame that clearly shows the field of interest. Other frames will be aligned to this reference frame. After that, go to Plugins, Stacks, StackReg. Try each of the different transformations to determine what is most effective. For each site imaged, factors that affect the detection of calcium transients will vary. By measuring certain properties, particle parameters, of each site, the sensitivity and specificity of detection are increased. Thus, for each site, we will calculate threshold for subtracted, threshold for ratio, and cell edge threshold. To begin, go to Plugins, Macros, Install, and select jncto.08release.ijm. Then, go to Plugins, Macros, Load Stack. Select a series and calculate the following particle parameters. To calculate the threshold for subtracted, select a rectangular region of interest over what you believe to be background. Measure. Note down mean. To calculate the threshold for ratio, select a rectangular region of interest around an optically flat area of the smooth muscle cell or cells. Measure. Note down mean and standard deviation. Repeat the process 2 more times, or you can draw 3 boxes at once by pressing Shift. Calculate and note down the threshold for ratio, calculated as the standard deviation divided by the mean, add 1, and then multiply the result by 32, averaging values from the 3 replicates. The factor of 32 is necessary to account for the ratioing algorithm, where no change from the preceding frame is mapped to an integer value of 32. This gives a good dynamic range to detect increases and decreases in the ratio for an 8-bit image. Select Image, Adjust, Threshold. Select Black and White. Define under the upper slide limit and note down the corresponding value. This is to the right of the slider. Only events occurring in the black regions will be counted. Click Reset. Now that the threshold for subtracted, threshold for ratio, and cell edge threshold have been calculated, we can analyze the image stacks. Determine the location of the stacks to be analyzed. Select just one series at this stage, that is, the first file and last file should be the same. Then select a location for output of data files and stacks. For example, data folder, output. Enter values of threshold for subtracted, threshold for ratio, and cell edge threshold. Leave other settings at their default values. The algorithm will produce a dual pane window in which the processed series is shown on the left and the original on the right. Open this file from the output folder and carefully go through this to assess the number of false positives and the occasions on which events visible by eye have not been detected. For each series, the algorithm produces an Excel file that details characteristics of the detected event, such as amplitude, area, and position. False positives, identified by reviewing the image files that the algorithm outputs, may be removed by hand from this Excel file, although you should have strict criteria for doing so. This is a movie of calcium transients in smooth muscle. In this example, the movie is comprised of 100 individual frames acquired at 5 Hz. We'd acquire many of these movies per experiment when investigating, for example, the effect of a particular drug on the frequency of calcium transients. Here, we see numerous spontaneous calcium transients in the smooth muscle cells. The outcome of the particle detection algorithm is shown here. For each series, the algorithm produces an Excel file that details characteristics of the detected event, such as amplitude, area, and position.
So we've demonstrated a method for imaging calcium transients in smooth muscle organs such as the urinary bladder. And we've demonstrated a way in which we analyse those calcium transients using an algorithm that we've produced in the lab. The important things to remember are to prepare the tissue well for the calcium imaging. So in the case of the tissue that we demonstrated today, that's removing connective tissue and removing any fat that may obscure the field. It's also important to pin the tissue in a flat sheet. Well, good luck with your experiments and thank you for watching.